The Dragon cargo ship, which arrived at the station late last month, carried opals and another large payload in its unpressurized section, its trunk. It also carried many other science payloads inside the freight carrier. A number of new experiments that have come to space designed to take advantage of the low Earth orbit environment. One of these new experiments is called Biotube Micro, and it's focused on how plants sense and respond to gravity. The principal investigator, Dr. Carl Hazenstein, joins us this morning from his office at the University of Louisiana at Lafayette. Dr. Hazenstein, people have been looking at how plants do in space for a long time. Generally, how do they do?
Well, generally they do quite well, especially once they get exposed to the proper environment. Initially, we did not know what it means to grow plants in the absence of gravity, but it turns out it wasn't gravity that was the culprit in not succeeding with plant growth. It was the lack of convection, and as a result, the distribution of gases and so forth that prevented proper development. But we have come a long way and learned how to do it in the meantime, so we can grow plants quite well.
The gravity is still a factor, right? What caused you to be focused on that aspect?
Well, it's a conundrum for more than 100 years to find out why every tree grows upright and anchors itself in the soil. And this basic question as to how tiny cells that don't have specialized features such as we have in our vestibular apparatus are able to sense and respond to gravity is just puzzling. And one of the aspects of detecting how these things work ideally requires to remove gravity as a factor. And this way we can gradually test how individual components of this system that makes plants sense and respond to all kinds of stimuli respond to gravity, and that's what this experiment is about.
Okay, take us through your plan then. What are you going to be? What have you sent to space, and what are you going to be doing in that hardware on orbit?
Well, the Biotube Micro is an external shell that contains the actual experiment. And the actual experiment consists of so-called magnetic chambers. And within these chambers, we have a series of exactly 10 magnets that are aligned so that they generate a very strong magnetic field. Inside this field, we distort this uniform magnetic field by, you could call it magnetic lenses. They're in reality ferromagnetic wedges. And what does that have to do with how plants respond to gravity? These magnetic gradients that result by inserting these wedges into the uniform magnetic field exert what we call a magnetophoretic force. And that force is specific for diamagnetic compounds, such as the amyloplasts that are the suspected gravity sensors.
It sounds like you're manipulating the gravity pull on these parts of the plant.
That's correct, but we don't exert a pull, we exert a push. These high-gradient magnetic fields repel these starch-filled amyloplasts, and by doing so, they produce the same effect that we see under the influence of gravity without having any other cellular component exposed to gravity. So we can pinpoint whether the amyloplast is actually doing the initiation of the sensing and response to gravity by virtue of moving them around in their respective cells. We call them statocytes.
Very interesting. Do the crew members participate in this in some way?
Not really. They were helpful in stowing the hardware into the express rack on ISS, but the actual experiment is entirely controlled from KSC.
Okay. I assume then that you've flown this on Dragon, you're going to get these samples back. What do you do with them when you get them?
Well, we will analyze different things. One is the distribution of these amyloplasts. After we have seen downlink video to indicate whether we observe actually curvature in the roots. So we have a video equipment that takes pictures in the infrared region of the spectrum, and we can see if the plants, if the roots curve. When we come back, we will correlate the distribution of the amyloplasts within these statocytes to the observed curvature, and we will also do a series of genetic tests to see if gene expression has changed in the growth conditions that we had in space. At the same time, we do so-called immunocytolocalization of relevant proteins that are responsible for the growth response of cells or roots in general.
So there's still plenty of study to be done once you get the samples back.
Yes, indeed.
Now, I found it very interesting that I saw that you ran this experiment on the last flight of Columbia, and were still able to get some results back from downlinked images, right?
Yeah, that's correct. It was unfortunate that we could not get our biological material back, but the downlink suggested strongly that we had curvature as we had expected, but we were not able to confirm this because we never received the actual plant material.
Is your goal to simply improve way plants grow in space, or does this have applicability on Earth?
Well, anytime you have a better or deeper understanding as to how plants perform and respond to environmental cues or problems, you are bound to benefit processes on Earth as well. So while we clearly want to establish a means of directionally controlling plant growth in space, we also want to use that gained knowledge for improvement of plants on Earth.
Dr. Hazenstein, thank you for taking a few minutes for us this morning, and good luck with the experiment.
Thank you so much.
Carl Hazenstein is the principal investigator of BioTube Micro.